Hello, welcome everyone. I would like to first of all thank you for our audience to join us this afternoon. And I would like to thank you, Amanda Minton, for accepting our invitation to attend this webinar together myself. I would like to also thank you, Swine to help us to develop this webinar. This is our third edition. And let me introduce myself for who doesn't know me. My name is Jean Pedro, or Jean, as I get used to be called here in the US. I am a Brazilian veterinarian. I work for MB Swine Reproduction, which is a joint venture between Magapor from Spain. Magapor is one of the leaders in the artificial insemination in the world, and Bretanha, our our parent company from Brazil, has a partnership for several years with Magapor in Brazil. And today with me we have Amanda Minton. She is an associate director with reproduction technology in Acuity Genetics. She earned her bachelor degree in animal science from Purdue University and her master's degree with swine reproduction from the University of Missouri. And today she has the oversight of Acuity board studies and supports their customers. And why we came together to this webinar? So we believe that our company has the same vision of reproduction and genetics. The main goal for this meeting and this webinar is to talk about genetics and genetic efficiency and tools to improve genetics to get opportunities from reproduction. MB Swine truly believe that the main reason for reproduction is to get more piglets from the best boars. Thus, we can spread genetics much faster. Good afternoon, Amanda. And first of all, I'd like to do the question. Would you agree with this sentence? And thank you again for joining us.
Yeah, well, hi, Jean, and hi, everybody. Once again, thanks, Jean, for having me. It's a pleasure to be a part of this webinar, and I think it's going to be a great conversation, and I hope that the audience feels the same way. But when we talk about genetics and reproduction, they really go hand in hand, and reproductive tools, both on the male and female side, are really a conduit to increasing genetic value in pork production systems. At the base of that, though, you've got to have a foundation, a good genetic foundation that's fully functional. And you can liken that to a toolbox, for example. So if our genetic platform is a toolbox, it's got to be free from holes and be rust-free, so it can facilitate the tools that we use to help express that genetic potential. And so I think reproductive tools and technologies can certainly expedite genetic improvement, but we've got to use it strategically. And for boars in particular, the right boars that we have in this stud, the index value of the boars, the environment that boar is in, all of those things, create value for the customer.
For sure, makes total sense. And we'll be discussing more about these tools and technologies more in details here. But before we enter in the main topic, I would like to bring a curiosity here. We made a calculation of what would be the financial gain for a system of 100,000 sows if they worked with 3 points more in EBV in average in their semen doses, meaning if they work with 100 EBV points today and tomorrow they start to work with 103 points, the gap here is like the calculation would be 100,000 sows multiplying by, let's say, 25 hogs per sow per year and multiplying per 8 cents on each EBV point, multiplying per 3 points. So that would be $600,000. So that's the gain that we've been talking about here. And we divided our webinar this afternoon in three main topics. And within these topics, we divided in three main tools. So the first topic is from the boar stud perspective. The second one is from the production perspective. And the third one would be from the sow farms perspective. So from the boar stud perspective, the first main tool that I brought to the table, it's the interval between collections. And in my personal opinion, this is a very important tool to get more from the genetics from the reproduction. So we know that the boar replacement in studs has been over 80 to 100% nowadays. And so the population in the boars are getting younger, right, Amanda? So how do you see this?
Yeah, so the replacement rate is really driven by and is dependent on the goals of the boar stud and the production system that they're within and are servicing. And so, we have two very broad goals when we think about boar studs, and one is high throughput and one is high indexing, semen. And so when we think about the nucleus level, of course, increasing genetic value is paramount. And so for those animals in particular, yes, we're going to target over 100% replacement rate because we really want to drive that genetic value. On the other hand, if you're a retail boar stud and are really about servicing commercial producers, you care a little bit more about high throughput. But at the end of the day, if you look at the average age of the boars in stud, it's really around 18 months. So yeah, we're working with a relatively young boar population in most of our studs.
Okay. And we know that few countries are already working with more collections in those young boars, decreasing the interval from 7 to 5 days to 3 to 4 days. And I brought a few graphics here from our study conducted in Brazil from Bretanha Suínos. And let me share my screen here to take a look in these numbers. So here, this study was conducted in a Bretanha's boar stud. So Bretanha has 4 boar studs in Brazil. And in this study, we divided the boars in 3 groups according to their age. And what we found out, it's what we were expecting. If we decrease the interval between collections, here the IC3 means 3 days of interval of collection, and here IC5, for example, is 5 days. You see that even if you decrease volume and decrease the concentration of the pure semen, by the end of the month or by the end of the year, you're gonna have more doses from these boars, and that happens in all the groups. Doesn't matter the age of that boar. So you could see here the older boars, for example, the third group, they showed in the end of over 800 more doses from these boars. So it shows a big gap here, right? So but even though these numbers, that there is still some systems or commercial board studs, I understand that the focus is a little different, but even there's some systems that work in the, let's say, old style, collecting every boar just once per week. And if they collect more the old boars, which in terms of genetic does not make a lot of sense, right, Amanda? Would you agree with that? What do you think about that?
Yeah, Gene, I agree. And I think that, your collection interval is really driven by dose demand a lot of the times, right? So if you got a stud has a lot of extra boards for contingency or whatever, it's really hard to justify the labor to continually increase collection frequencies on some of your boars. On the other hand, if you've got a boar stud that's sold out of doses and we're in the middle of the summer and trash rates increase, we know that those collection frequencies go down to 3 or 2 days. But I think consistency is key, and I think the data that you showed would probably say that as well. But the consistency is the part that I think we need to focus on, because your data and data from Billy Flowers from several years ago, would show that we can collect boars that are boars 2 times a week and show no detrimental impact on semen quality, but the key is consistency, because he also showed that if it's inconsistent, so 3 days rest 1 day, 7 the next, 3 the next, that we will see some short-term implications on semen quality, and even long-term, a portion of those boars will never resume the semen quality that they once had. But if you're consistent in that collection interval, to your point, you're going to get a lot more use out of those young boars that you have in stud.
Sure. And so, and just to wrap these two, would you explain to our audience why it's so important to get more doses per boar per year?
Sure. Yeah. I think, the crux of that question goes back to just utilization of the boars that we have in stud. And so being able to get more doses out of a single boar, and use that boar for more matings within your system. So if you can decrease sperm per dose, or you can get more doses out of a boar, then we can essentially manage that boar inventory a lot differently. And we don't need those bottom third boars, for example. And then that actually does then increase or decrease our selection rate from the boars that are coming in. So, that part of it, ultimately, the boars that are in stud have a profound impact on the system through the traits that they possess, and then that carries through to the market pigs and ultimately, the customer's pocketbook.
Sure. Okay, jumping to the next tool here that we brought is the management of animal collections, board collections by EBV or by genetic index. It's very correlated to the last tool, but it's different. Do you think this is an opportunity? How do you see that, Amanda?
I do, and I think for Acuity, we've got some boards specifically for one of our large customers, and they specifically want semen distributed, by index to their most productive farms. And I think intuitively and economically that makes perfect sense, because the sow farms that are most productive are going to produce the most pigs, and those pigs are most valuable. So I think there's definitely some opportunity there within the U.S. industry, and would encourage producers to work with their boarsa and their genetic company to really optimize what they're receiving.
Yeah, that would be very impactful for sure. And with this tool, I brought here an management that Bretanha in Brazil again is working very hard in one of their customers. So this is data again from a Bretanha board study in Brazil that they are managing the EBV per dose per month. You can see along the time here since January of 2020. Very nice ramp up here, and if you divided these EBVs per quarter, you would see a very interesting ramp up as well. So that means a financial increase here for sure. And how do you see these, Amanda, meaning still a gap, right? Still something to work before that.
Yeah, I think so. I think there's some opportunity in how we strategically use boards with our customers. And the main thing that we want to make sure happens though is what we're sending, the quality of the semen, and I guess the index value of the semen is actually being realized at the commercial level. And so when you think about that's really the key, right? Is what we think that we're doing from a genetic standpoint is actually being seen from a commercial standpoint. I think that's why, like a commercial test herd, for example, or a CTH is important to improve that genetic process, so that we use the CTH to make single sire matings, and at a commercial sow farm, then that feeds that data into the genetic program, so that what we're measuring is actually being seen on a commercial perspective.
Sure. Okay. That makes sense. And the third tool here that we brought is nutrition and water quality in a boar stud. I think this is a topic that we still do not have a lot of research, right?
Yep.
How you've been conducting these in the Acuity studies?
Yeah, so from an acuity standpoint, I'd say we're kind of in the process of deciding what our strategy is going to be going forward from a nutrition standpoint. Like you said, there's not a lot of research out there that's available, and so really what we're trying to do as an industry is just make sure boars are not deficient in protein and energy. The challenge comes in when you think about micronutrients and when and where you add those in. And what kind of impact are they going to have on semen quality? And there's different papers out there, but a lot of it's inconsistent. So do you need DHA in your diet? Do you need phytogenics? There's all these different things that could improve semen quality, but trying to really understand what's the most impactful in the boar diet is hard to do. And that's just on the mature boar side, right? The pubertal boar, and there's a lot of work that needs to be done, boars that are much younger than that from the development.
Sure. No, and that's a message for our research audience. Maybe we can conduct more research in this direction in nutrition. And okay, and skipping and jumping from the second topic here, main topic, it's from the production perspective, how to get opportunities from this part. And here you've already mentioned about single sires, producing those single sires against producing those with pools, which you mix 3 or 4 boars or even more boars in the same solution. We know that there are many countries out there that are working with single sires, and I think you would agree with me. There is a big gap for us to capture here. But what's your opinion? How we can move forward to this step of working more with single sires?
Yeah, it's a great question, and I think it's a debate within the U.S. industry at least. Because single sire matings have a very negative connotation in our industry for one reason or another. It's primarily because we've all used pooling as a protection mechanism. And yes, we have poor fertility boars in studs. We know that. And estimates are around 20% of boars are subfertile. But on the other end of the spectrum, we have some really highly fertile boars that are not expressing their potential because we're pooling them. And so if you look at some of the research, again, Billy Flowers and some others that have done, and just looked at the pools, they found that a single boar, can sire 65% of the litter in that pool. And so, we're really not capturing the value by using pools. And I know that's hard. It's hard for people to hear because we've always thought about it as a protection mechanism or insurance. But in order for us to really move the needle on reproductive performance and capturing that genetic value, we've got to really start thinking about using single sire semen as opposed to pooled.
Right. No, that's I agree 200% here. That's a main gap for our industry in US, I think. The second tool here, it's more about prevention and not working direct to the tool, but it's semen doses analysis. And right here, I'd like just to introduce saying that there is 2 different types of analysis: the qualitative and the quantitative analysis in the semen. Could you explain to our audience what's their difference?
Yeah, so when I think about that, we think about just the sheer number of sperm that are in the dose, and there's a lot of different research that looks at what that optimum is. So you've got the number of sperm that's in the dose versus the actual quality of the dose itself. And so those are kind of the 2 differentiating factors in terms of what we're looking at from a boar stud perspective.
Okay. And what's the analysis that have been performed in a daily basis in Acuity studies?
Yeah, so we do use CASA systems at each of our studs, and we use those for consistency and accuracy. We've got some great people that work at our boar studs and do a great job from a manual perspective, but we all know that the days get long and you're collecting boars at 3 p.m. when you started at 3 a.m. And so just having a CASA system in place just helps tighten that gap when it comes to sperm produce and consistency with motility and morphology.
No, I definitely agree as well. And so, I know you also perform a third part analysis in a third part lab, and so why do you do that?
Yeah, we've really ramped up our third party evaluation over the last few years. So we went to sending in samples every few weeks to actually sending a sample from 50% of our batches on a daily basis. And really that helps us understand where we're at and if we need to make adjustments to things over time. So it gives us a really good picture of what we're sending out the door. There have been instances where the third party results would tell us something differently than what we believe we sent out the door. And so in those instances, it gives us a chance to replace some of those batches at the sow farms to make sure that we are getting as good quality semen as we can, to our customers.
Okay, yeah. No. So, and how about the difference between you also have to perform an internal quality control in your studies comparing to what you perform outside. So why it's so important to also perform a internal quality control?
Yeah, so we do motility checks, I think like a lot of boar studs do, but internally just to make sure that motility doesn't go below what we would like it to. And we do that just to kind of compare with what we've seen at the third party lab because we know we are shipping those samples to the third party lab, and just with UPS and temperature changes, the motilities may not be exactly the same. And so it's important to understand also, what we're getting at the stud level for that semen that hasn't been through that transport process. So that's where I think that piece is important too. And if we could, Jean, I have the graph on looking at semen quality and fertility. If we could pull that up, I just want to just show the audience. I'll wait till you pull it up. So this graph, this graph looks at conception rate versus percent motile and normal. And the reason I think this is important is because it shows a picture that the semen quality that we measure at the stud is not always equal to fertility. It can look good based on our parameters. But as you can see in this graph, these blue lines are individual boars, and each of these individual boars have had at least 50 single sire matings. The gray line is average motility for those collections, and then the black is the average normal morphology. And so you can see we've got boars that are less than 30% conception rate, but the motility and morphology looks good going out the door. And so I think it's important to understand that semen quality, as we've traditionally measured it, is not always equal to fertility. And so that's why one of the other things that we've been working on that kind of goes along with this, Gina, is working on a model to try and predict boar fertility, in that we're measuring, we have a lot of single sire matings that we have, and that we're looking at some different assays, so different functional components of sperm cells that are actually influencing the fertility other than your traditional motility and morphology measurements.
Yep, notes. Pretty interesting graph, Amanda. And you mentioned also about the CASA, and I think it's important to understand. For example, we know that the CASA system can help us to standardize the semen those results and close up that results through its technology that taking out the human factor from this production. Or how now, right? So how do you see this allow us to work with lower concentration? How do you see the CASA in this topic?
Well, I think as we push sperm per dose lower and lower, and I think we will get to that point, like you alluded to earlier, there's some other countries that are using a much lower sperm per dose than we are today and getting exceptional reproductive performance. So, but I think the important part and where the CASA system comes in is that as we push that lower sperm per dose, we need to make sure our precision and accuracy is very tight, and to make sure that doses that we're sending out the door that are 1.5 billion are not 0.8 billion, right? And so it helps from a precision, like I said, a precision and accuracy perspective. And then I also think, as you look at that over time, you can make adjustments if you need to, but I do think it helps from that perspective.
Right, and you mentioned about a few other countries that work with lower concentration, and I brought a few numbers here. For example, in Holland, it's one of the most aggressive countries in terms of concentration. They are working with 1 billion total for post-cervical doses and 1.8 billion for traditional. And in Brazil, again, Bretanha studies are working with 1.25 billion for post-cervical and 2.5 for traditional. Spain work with 1 billion and 2 billion, but viable cells. So how do you see this improvement in concentration going further in US? And how about in your system? What's the concentration that you are working with?
Yeah, so in our system, all of the traditional or cervical AI is in it with 2 billion today. And so about 10 years ago, we did a titration study to look at what sperm per dose is optimal for reproductive performance using just regular AI. And so our low is 1 billion up to 3.5. And through that study, is a very large study, we would determine that 2 billion total sperm per dose was optimal to maintain reproductive performance with traditional AI. So we've done several studies over the years on traditional and PCAI. PCAI today, we're at 1.5 billion total. And so we probably still have some more work we can do there, but that's where we are today. And to your point, really the driver behind all that, the driver behind decreasing it to 2 billion and 1.5 for PCAI came back to the bore studs and making sure that we were utilizing the best bores that we could in the studs and really spreading those bores out to more meetings.
Okay. Yep. And the third tool here in this topic is it could be a more controversial, and I think everybody would agree very more challenging. It's the one delivery per week. So delivering semen doses to the sow farms just once per week. And because of the winter and all the transportation issues that we have, do you think this is already a reality, Amanda? How do you see this?
Yeah, so I'd say it's not a reality today. And actually, the last couple of years, we've kind of gone backwards from that perspective. So ourselves, I know others in the industry have gone to 3-time-a-week delivery as opposed to 2-time-a-week delivery. And that's really been a request of our customers because there's a lot of questions or concerns around semen age, and in particular, semen age and a lower sperm per dose. And so I think that's really why, we made the switch to that 3 times a week. So I think to get to 1-time-a-week delivery, we're going to have to prove to our customers that the semen quality is just as good, and we're not going to see any kind of dip in reproduction. I mean, the other consideration is, what are the pros and cons from a bore stud operational standpoint? So, one of the pros I see to that, Gene, is biosecurity. So fewer touch points at the bore stud, fewer touch points at the sow farm. So that is certainly an advantage to 1-time-a-week delivery. I guess one of the cons to that is I think about our bore stud teams and just collection frequencies and just, does that really concentrate your collections on a couple days? And if so, how does that impact your labor? How does that impact the people that work there? There's already pressure on them to get a certain number of doses out at a certain time every day. And so how does that impact that? I don't know exactly. So I think it's an intriguing question, and one we have to think about more.
Sure, sure. No, it's for sure still a few steps until we get that point, but it's for sure it could be an opportunity and a tool to get more from genetics for sure. And just to wrap up this topic, you mentioned about the importance of guaranteeing the semen quality, and we receive a lot of questions from the farmers, from the customers about semen age, and what's the importance of the extender to make this tool happen in maybe the future.
Yeah, I think the extender is critically important because as we've looked at extenders over time and just looked at how far extenders have come in terms of semen preservation, I do think that's really important in the protection of the sperm cells during storage. So it's certainly a piece to consider.
For sure, for sure. Okay. And now, starting the third topic here would be from the cell farm perspective. How can we get the opportunities from this part of the chain? So then one of the main tools here, we've already mentioned about that, it's the post-cervical artificial insemination. And I brought a number that around 45% of the sows in US are being bred using the post-cervical. How about Acuity? How about Maschhoffs system? What's this number today?
Yeah, so within the Maschhoffs, 100% of our sows are about that, right? Except for opportunities and that kind of thing, are used. We use PCAI on those sows. We've not done a lot of work with gilts to this point, but I certainly think PCAI is a valid option in gilts. But we are utilizing PCAI in all of our farms. I think one of the mistakes that we made when we first rolled PCAI out to the farms was really overestimating the labor savings. Yeah, I think there is some there, but really the true value and the purpose that you should use PCAI is just for that genetic value, that genetic gain. And so if you're not doing that, if you're not using a lower sperm per dose and seeing higher overall index in your semen, then you might be spending some extra money on a catheter that is not creating a big return. So I think that's the primary reason to use it.
Sure, and that's for me does not make sense for that I see outside there people using post cervical but with a full dose that make me crazy. So and about there is few new technologies regarding post cervical as well, and one of them is the bulk bags that. We see an increase in the use of this technology, especially here in the US. And how do you see this topic?
Yeah, so we have used bulk bags, and there for a while, about 80% of our sows were on bulk bags, but we've since transitioned back to individual doses. And if you wouldn't mind, Jean, that second graph that I sent that looks at the multi-dose bag semen concentration, but what we found over time, and so we did use two different size of bags, and so this graph is just for the 70 dose bag. And so for the specific multi-dose bag that we were using, we just found over time, whether or not you mixed the bag or not between the first and the 70th dose, we see a steady decline in the sperm per dose that we're actually delivering to the sow. And so that was concerning for us in particular. I know there's other multi-dose bags out there now and some that may agitate the semen and mix it up, but from our experience, that decrease in concentration over time kind of told us to go back to individual doses and to put that quality controlled piece back at the boar stud rather than having the sow farms mix that to make sure that they get the right semen dose in the sows.
Interesting. Yeah, I in my personal opinion that increase a little bit more the risks, and in the standpoint of the labor as well. And plastics, that's another whole story that we talked about in our second webinar. But let me jump for the next two here, and this is about to fix the time artificial insemination and the possibility of reducing the number of doses per sow per cycle, maybe giving that one very accurate shot in that female. Do you think this will be a tool to improve the genetic efficiency, Amanda? What is the latest studies about this topic?
Yeah, so I believe that single fixed timed AI can be a great tool. And so we've done several studies over the years. When JBS United first, or United Animal Health first came out with Avigel, we did some of the initial trials, and then since then have done some more trials with that product. And so, throughout the most recent trials, we didn't see any difference in conception rate, bearing rate, and litter size between the Avigel treated sows and our controlled sows. Now, I say all that, and today we're using it on very few sows, only because it's hard to implement. It's hard to execute. So I think it's a great technology. I think it's very useful. But the execution piece and getting that timing right is really important, and is not something that we can always get done on the sow farms. We just have so many other things going on, and we can't get that timing down quite right. But I do think it's a really great tool.
No, for sure. Yeah, if you have the possibility to use always the same board for always that group of sows, that's an amazing tool that we would have in hands. And the last tool in this topic here, it comes together. The last one that we just spoke about, it's the replacement rate in sow farms, and this is a little bit about the research that we've been trying to develop in Brazil about creating subpopulations in the sow farms. We know that nowadays over 50% of the sows has less than 3 farrows in the sow farms. So if we could potentially use the best EBV doses for those young sows, that would speed up even more the genetic gain in these farms. And perhaps in the near future, we could provide an EBV per piglet. That would be such a nice thing. So what do you think about this, Amanda? Is that too far? Is that a next step? How do you see this too?
Yeah, I certainly think that you could target certain populations on your sow farm with the best indexing boars. I mean, I think what that would take is that you have to actually know the index value of your gilts coming in, and you have to know the index value of the sows that are in your herd. We're going to make the assumption that the index of the gilts coming in should be higher than the sows that are on your farm. So I think it kind of depends on what the index differential is. And at the same time, though, I think you have to balance that with, the reproductive capacity of your sows. So, I think you maybe target your gilts, but then also some of your P3 through 5 that are the most reproductive, depending again, like on the index difference, that are the most reproductive females on your farm. I think you could think about different ways that you could do that on your farm for sure.
Okay. Yep. No, I agree. And I think we touched the three perspectives that we'd like to touch here. Going a little further for future and last couple of questions here. And how do you see the swine reproduction and genetic walking through the future, Amanda?
Yes, I think there's a lot of new technologies out there. There are a lot of things that are being researched, such as using cameras and thermal imaging and saliva samples to detect estrus and ovulation. So I think those types of technologies are something that proven. We need to learn how to embrace and how to use them in our operations. So those are some things that I think are coming down the line and that I think will be exciting. The other piece that I think we need to think about is a little bit I alluded to earlier, but just nutrition of the sow. during lactation, and then mid-gestation and beyond, and what that does in utero to our replacement boars and gilts. I think we need to understand not just the economics of that feeding program, but what that actually does to the physiology of our replacement gilts and boars as well that we're bringing into our farms.
For sure, for sure. In a standpoint of a boar stud, how do you see a boar stud in the future?
Yeah, so from a boar stud perspective, there's a quote that I like that's from a well-known children's book character, Winnie the Pooh. And the quote is, "I always get to where I'm going by walking away from where I've been." And so I think the purpose of that quote is, I think that on the boar studs in particular, we kind of need to walk away from what we've done the last 20 years. And we need to maybe walk away from some of those old paradigms and kind of shift our mindset, because I think as the U.S. industry in particular, and contemplating, single sire versus pooled, I don't think that we are maximizing our reproductive potential and optimizing just that genetic value. And so I think, we're at a point where we need to critically assess where we want to go. And if we're happy being average, being status quo, being comfortable, then we don't need to make any changes. But I will challenge the group and say that if we want to be exceptional, if we want to compete on a global scale, then I think we're going to need to make some changes and be uncomfortable. So I think from a boar stud perspective, things will get more complex as we evolve. And I believe that we will manage a boar on an individual basis rather than a pool. I think that will become critical. And then I think that we'll be able to measure some things in the stud real time that we can't today. So I think we'll be able to measure things like DNA fragmentation, acrosome status, link signature, some of those other functional things that help actually determine boar fertility real time. And that helps us make a decision on an individual boar about, are we going to use that boar? Are we going to use them at 3 billion sperm per dose? Are we going to use them at 1 billion sperm per dose? So I think that part of it is really exciting, Gene. And we just have to be prepared to train our people that we have and then recruit people that are more technologically savvy maybe than what we have before. So I think it's an exciting time from a reproductive perspective.
Well, that makes total sense. And I think that wraps up our conversation very well in terms of how do you see the future using more these tools and really breaking these paradigms. And from my end, I would like to leave a message as well here, and because the result, I would like to say that the results of 2022 have already started, and last week the reproductive results. So actually on the last 9th of September, the first inseminations of January 1st were done last week. So if you want to change your results from 2022, just start today. If not, you're late. Amanda, thank you again for the opportunity. Thank you for being with us, and I really appreciate our conversation. And let's move forward to the next steps in genetic efficiency.
I appreciate it, Gene.
Thank you, everybody, and we talk soon.
Bye-bye. All right, thank you.